Welcome to this lecture on the mechanism of reproductive hormones. Today, we're going to explore how tiny chemical messengers in our bloodstream control the very foundations of life, from puberty to pregnancy. By the end of this session, you will understand not just the biology, but the underlying mathematics of how these hormones interact with our cells. To begin, imagine you are in a crowded, noisy room where everyone is shouting different messages. If you have a walkie-talkie tuned to one specific frequency, you will only hear the message meant for you, completely ignoring the background noise. This is exactly how cells in our body communicate. Reproductive hormones travel through the bloodstream, essentially shouting instructions to the entire body. However, only specific target cells equipped with the right receivers, which we call receptors, can actually hear and respond to these hormonal messages. Now that we understand the basic analogy, let us look at the formal biological definition. In reproductive biology, the mechanism of hormone action refers to the specific biochemical pathways through which hormones like estrogen or testosterone exert their effects. When a hormone reaches a target cell, it must bind to a highly specific protein molecule known as a receptor. This binding event is the crucial trigger. It sets off a cascade of cellular events, which might involve activating enzymes near the cell membrane or even entering the nucleus to alter how specific genes are expressed. This brings us to an important distinction between the two main types of reproductive hormones. Peptide hormones are water-soluble. Because they cannot pass through the fatty cell membrane, they bind to receptors on the outside of the cell and rely on internal second messengers to get the job done. In contrast, steroid hormones are fat-soluble. This allows them to slip right through the cell membrane and bind with receptors inside the cell, directly influencing the cell's DNA. The chemical structure of the hormone completely dictates its mechanism of action. Before we move on, let us see this with a concrete visual example. On the left side of this diagram, you can see a peptide hormone binding to a receptor on the outer surface of the cell membrane. Notice how it triggers a second messenger inside the cell to carry out the response. On the right side, observe the steroid hormone. Because it is fat-soluble, it passes directly through the outer membrane, binds to a receptor inside the cell, and travels straight into the nucleus to interact with the DNA. This visual perfectly summarizes the two distinct pathways. Understanding these cellular pathways is not just academic. It is absolutely crucial for modern medicine. Without these precise mechanisms, our bodies could not regulate puberty, maintain a pregnancy, or produce sperm and eggs. In the real world, this knowledge is the foundation for developing birth control pills. These pills use synthetic hormones to trick the body's receptors and prevent ovulation. Furthermore, treating conditions like infertility or hormone-dependent cancers relies entirely on our ability to block or stimulate these specific hormone receptor pathways using targeted drugs. To truly understand how strongly a hormone affects a cell, we must look at the mathematics of hormone receptor binding. The biological response is generally proportional to the number of receptors currently bound by a hormone. We calculate this using the fractional occupancy formula. Y represents the fractional occupancy, which is the percentage of bound receptors. H, in brackets, is the concentration of the free hormone. Kd is the dissociation constant, which tells us the hormone concentration needed to occupy exactly half the receptors. If Kd is high, the hormone has a low affinity, meaning you need a lot more hormone to get the same biological effect. Now that we have our formula, Let us apply it to a real scenario involving luteinizing hormone on the Leydig cells of the testes. We are given a dissociation constant of 4 nanomolar and a current hormone concentration of 12 nanomolar. We want to find the fraction of occupied receptors. We substitute 12 for the hormone concentration and 4 for the dissociation constant into our equation. The numerator is the available hormone, and the denominator represents the total binding dynamics. Dividing 12 by 16 gives us 0.75. This means 75% of the receptors are occupied, sending a very strong signal to the cell to produce testosterone. This mathematical relationship perfectly mirrors biological reality, which brings us to a clinical application, comparing steroid hormone affinities. Imagine a pharmaceutical company testing a new synthetic estrogen for birth control. The natural estrogen in the body has a dissociation constant of 8 nanomolar, while the new synthetic estrogen has a much lower dissociation constant of 2 nanomolar. If a patient has a circulating hormone concentration of 8 nanomolar for both substances, we need to calculate and compare their receptor occupancies to see which provides a stronger biological signal at the exact same dosage. Let us first calculate the fractional occupancy for the natural estrogen. We are given a hormone concentration of 8 nanomolar and a specific dissociation constant of 8 nanomolar. We plug these values into our formula. The numerator is 8, and the denominator becomes 8 plus 8, which is 16. Dividing 8 by 16 gives us exactly 0.50. This makes perfect mathematical sense. Whenever the hormone concentration exactly matches the dissociation constant, the receptor occupancy will always be exactly half, or 50%. Now let us see how the synthetic version compares. Now we perform the exact same calculation for the synthetic estrogen. We use the same hormone concentration of 8 nanomolar, but we substitute its lower dissociation constant of 2 nanomolar. Setting up the equation, we get 8 divided by the sum of 8 and 2. This simplifies to 8 over 10, which yields a final result of 0.80. Because the synthetic hormone has a smaller dissociation constant, it mathematically shrinks the denominator, driving the overall fraction higher. Physically, this means 80% of the receptors are occupied, exerting a much stronger contraceptive effect on the body, even at the same dose. To wrap up our lecture, let us review the most important ideas we have covered. First, reproductive hormones act as chemical messengers that only affect cells with specific matching receptors. Second, the chemical nature of the hormone dictates its path. Peptide hormones act on the cell surface, while steroid hormones act directly on the DNA inside. Finally, the strength of these biological responses is governed by the mathematics of receptor binding. A lower dissociation constant means a higher affinity, which is exactly how synthetic hormones in medicine are designed to be so highly effective. Thank you for joining this session.